Codominance is a non-Mendelian genetics. According to Mendel, law of dominance play a high priority. When there is a pair of contrasting character, one will be dominant, the other will be recessive. But in case of codominance, we will see that two dominance allele express equally and produce a new phenotype. Where do you see such an example? We see it in ABO blood grouping in man. In human, we have IA as a gene which expresses for A blood group. Then you also have IB, a gene which expresses for B blood group. You also have a condition where IA and IB comes together and forms a blood group called AB blood group. Now this AB blood group is called as codominant type. So we will see how this is formed. Now whenever we start a crossing, you should remember that we should start off with a homozygous form. So the homozygous parent today we are going to consider is of IAIA, that is blood group A, is being crossed with IBIB, which is of blood group B type parent. So the next step what we should do, we should take out gametes. So when we get gametes out, you get IA separately, IB separately. Then what happens? We are going to fuse them for F1 generation and you get is IA, IB. Now if I have to write down here, it is blood group A and this will be your blood group B. We started with blood group A and blood group B, but codominantly you can see a blood group newly formed, blood group AB has formed, where both A and B are codominant. Now the rest of the thing we can see that when we are selfing them, so we are going to cross with the same type IA, IB. So what do you call this process as? Selfing. When you are selfing them, you get the next layer of gametes in two parts. You have IA, IB separately and in this case again another IA, IB. So in this gametes 2 or otherwise I can say F1 generation gametes are IA and IB. So we will cross them, we will form a Punnett checkerboard. So this is the Punnett checkerboard. Here one comes from father, one comes from mother. So we are forming F2 generation. Now if you remember, the gametes what we formed were IA, IB from father and IA, IB from mother. Now what are the possible offsprings we see in F2 generation? You see the first one as IA, IA, second IA, IB, then we have IA, IB and you have IB, IB. So here you have blood group A type, you have blood group AB type, you have again here AB and here B. So when you are going to check for the phenotype, phenotypic ratio would be 1A, 2AB and 1B. So the ratio is 1:2:1. Then you have genotype. So genotypic ratio is 1 IAIA, 2 IAIB and 1 IBIB. So again the ratio is 1:2:1. So you can compare codominance and incomplete dominance have same genotypic and phenotypic ratio.